Okay, today we're going to talk about germline editing in mice with the CRISPR system. But before we get into that, we need to know something about transgenic mice in general and how traditionally they have been made. So we're going to go back about 20 years to the earliest ways in which people did genome editing in mice. Okay, so transgenic mice are those whose genomes have been altered by technological means. So the oldest way, which has been around a long time, which is now back in fashion for reasons you'll understand in a sec. Okay, we simply inject the intended transgene into zygotes. Okay, it was not really feasible to get knockout mice in this way, largely because there was no control over the location of the DNA that went in or the copy number. Say, the newer way uses embryonic stem cells, which has got issues of its own. It's not easy to get knockouts in that system, but it is possible. So the newest way uses CRISPR to inject CRISPR components into zygotes. Because the biology here is so much different from the earlier just adding a plasmid DNA, editing occurs very efficiently. This is a huge advance. And CRISPR also can be put into ES cells with similar efficiency. It's really huge. Okay, so the traditional way to make transgenic mice is with zygotes. So you start off with a donor female who's been treated with hormones to superovulate, and then she's mated to a male, and fertilized eggs are then collected from her oviduct the next day. Then the eggs can be deliberately injected with a microinjection system with the DNA that is to be taken up by the nuclei of the cells. Okay, so once that has happened, these cells are then incubated for a while and then transferred back into a pseudo-pregnant foster mother. So she's been treated with hormones to allow her to accept the injected eggs, and hopefully some of those will then be implanted and develop into mice. So once the baby mice are obtained, they analyze the DNA. They show this by little tail snippets. They actually do cut off the little end of the little baby's tail in order to test whether the intended transgene was successful and where it went in some instances. And notice that the babies are dark-colored fur versus the pseudo-mother who's white-colored fur. The fur markings are often used to ensure that the genotype, or the strain at least of the mouse, is what you intended it to be. Okay, so this is what I said. Donor mother superovulated, mated, zygotes recovered. And then for CRISPR, we would inject CRISPR components rather than the DNA transgene, and they will be cultured until the blastocyst stage, when they are then implanted into the uterus and get the baby mice and then genotype them. Okay, so the old problems are not a problem anymore, because if you just inject something like a plasmid DNA into a zygote, first of all, it doesn't guarantee the DNA is going to become incorporated anyway. But more importantly, DNA will not occur at its homologous location most of the time. So what will happen is you can get the DNA inserted at almost random locations in the genome and not necessarily in single copy. And it's certainly not going to have one copy at each of its homologous chromosomal loci. So usually there are many copies, and so the baby mice that you get are at best heterozygous for the transgene, and they must be bred to get homozygotes. Knockouts are virtually impossible because of their rarity. Okay, this has totally changed with the CRISPR system, because you can get your single-cell zygotes and inject them with the CRISPR components. Now, the CRISPR directs the guide RNA to the correct location in the genome in order to perform the editing. So this is very efficient in terms of getting the intended DNA to the place where you want it to be, or making the desired edit. So the efficiency of correct editing is really key here. So after you get your embryos, you can then transfer them to a pseudo-pregnant female, get your baby mice, and genotype them. So it's the same system, but it's the fate of the nucleic acid that's injected that is so different in this system. Okay, so it's also very efficient at making knock-ins like GFP and epitope tags, because you can manipulate the injected nucleic acid to basically include extra sequences that weren't there in the first place. Okay, other knock-ins include epitope tags, fluorescent reporters, conditional alleles, many, many options. So the fact that it's very efficient and very specific, although not necessarily totally specific, it edits the target locus to which the CRISPR components were designed. And it can easily be made to use knockouts, well, not as easily in mammalian cells as in some other cell types, but it can be done using homology repair templates, along with the Cas9 and the guide RNAs. Okay, we'll talk more about that in a minute. Okay, it's also incredibly amazing that the CRISPR very often edits both alleles in the diploid. So you don't have to do it twice to get one of each, or you don't necessarily need to breed. Baby mice that are homozygous for the desired edit will occur very frequently. Okay, so embryonic stem cells are also used, have been traditionally, and still are used in the CRISPR system. So what embryonic stem cells are, or ES cells, they're cells that are derived from the inner cell mass of early embryos. And they can be maintained, they're grown in culture, in such a way that they don't differentiate. But they retain their capacity to differentiate if they're reintroduced into a host zygote or blastocyst. So the reason that for their use is that it can be grown like any other cell line. They can be transfected or electroporated to get the DNA in, then grown up clonally and examined for the desired DNA event. So it may be random, partly, well, not really, but it could be random. But the point is that you can actually look for the thing you want before investing the effort in making a mice. So if you look for ones that are intended to be a knockout, and they are a knockout, you can get those cells, and they can be screened for, and then they can be used to transfer to a blastocyst to grow up the baby mice. So the initial manipulations are in the stems or in ES cells, where they can be grown and tested, manipulated, and then used to generate a mouse later on down the line. So that's basically showing what's happening here. The very early blastocyst embryo has an inner cell mass, and those cells were at one time harvested from the blastocyst, where they can now be grown in culture, manipulated, screened for the desired genetic changes, and then grown up clonally and then used to re-inoculate a blastocyst. Now, making a new mouse out of this isn't trivial, because these are cells and they have to be injected into another blastocyst in order to become part of a chimeric transgenic mouse. Okay, so envisioning what's happening here, we have embryonic stem cells. We've transfected them, and then we can grow them, and then we can select for, or identify at least, the rare targeted cell that we're interested in, acquire a pure population of those cells. But now to regenerate a mouse, those cells need to be injected into a host blastocyst, which is obtained from a superovulated mother. Okay, now those injected cells will actually commingle with the blastocyst cells and contribute to the formation of the adult tissues in the new mouse. So the new mouse has essentially four parents. It has the parents of the blastocyst, into which the ES cells were injected, and then it has the parents of the ES cells themselves. So the mouse you get is what's called chimeric, because it has genetically different tissues as part of the same mouse. And there's very vividly seen if you use mice of different colors, and I'll show you a picture in a sec. So what has happened is those ES cells have contributed to the formation of the adult tissues, and hopefully have contributed to the germline, so that when that mouse is bred, some of the ES cells will end up being the parents of the new animals. So I just said this: once the ES cells is obtained, injected into a blastocyst, implanted into a pseudo-pregnant foster mouse. Okay, and the important point is the baby mouse or pup is a chimera or mosaic of two different types of cells, some from the injected blastocyst and some from the ES cells. Here's a picture of some chimeric mice. The baby mice are brown and white because the brown cells come from the ES cells and the white cells come from the host embryo. So it's a very vivid way of looking at the fact that these are chimeric mice. Okay, so these must be bred to generate non-chimeric animals, And the fact of it is, ES cells must contribute to the germline, which it seems that they do. And then, of course, they must be bred again to get homozygous lines of mice. The amount of work involved is huge. Okay, so we're going to talk about a very early 2013 proof of concept experiment in which we're going to try to manipulate mouse genes by simply changing restriction sites just to make sure the CRISPR system works and to get a handle on how efficient this is going to be in these embryos and how manipulable the system is going to be. Okay, so the experimental system here is some TET genes which are involved in DNA methylation in mouse, and we wanted to make knockouts of these singly and in combination. Okay, so you can design targeting sequences, and they were very clever in how they did this. They looked for naturally occurring restriction sites within exons that were close to a PAM sequence. So these sequences have not been manipulated. They're naturally occurring. The TET1 gene has a SAC1 site, a few nucleotides upstream of a PAM sequence, which, if you remember, is NGG. It's for TET1. For TET2, there's a naturally occurring EcoR5 site, similarly adjacent to a PAM sequence, and similarly for TET3. So they were exploiting the natural restriction sites as a way to first to look for and then target and then analyze whether the desired genetic changes that they were going to make were in fact there. Okay, so these experiments are going to involve, first of all, ES cells, just to see if it's easy to do, it's possible to work, without generating a mouse, just looking at the transfection and the products of this transfection. Then they will do it by embryo transfer, which we expect to be repaired by non-homologous end joining. And then finally, we're going to do an experiment in which we will insert, as well as the targeting guides and the Cas9, of course, oligonucleotides, which will serve as homology repair templates to basically replace what's there at the locus with the sequence within the oligonucleotide. Okay, so culture ES cells, just to try it out, you're going to transfect the cell cultures with DNAs encoding Cas9 and their chosen guides, and then we can tell if it's worked or not by looking at the restriction site, which is going to be destroyed if the Cas9 nuclease actually worked, because the restriction site is within a few nucleotides of the PAM and within the guides, adjacent to the guide sequence, or within the guide sequence. So the TET1 has a restriction site of SAC1, GAG, CTC. TET2 has EcoR5, GAT, ATC, and TET3 has ZO1, CTC, GAG. And those are within the guide sequence and adjacent to the PAM. Okay, looking at this figure again, the underlying sequence is the guide, and we expect that if the CRISPR got in, did its job in cutting the DNA specifically adjacent to the PAM sequence, with the result that a few nucleotides are deleted as part of the homology, the non-homologous end joining pathway. So what we expect to happen in a successful CRISPR experiment here, the CRISPR will go in, it will be targeted to the underlying region, and it will cut the DNA a few nucleotides 5' to the PAM, which in this instance is right within the SAC1 site. Same with the EcoR5, that targeting sequence will cause a cut within the EcoR5 sequence, or very close to it, and the same for the ZO1. So the first thing we're going to look for is the loss of the SAC1 and EcoR5 and ZO sequences as a result of this manipulation, because we expect the DNA that will be cut with CRISPR, a few nucleotides will be lost in the vicinity of the cut, and when it ligates back together, those nucleotides will be lost, with the result that can be detected by digestion of the DNA with a restriction enzyme. So it's an easy test to see what happened, to see, first of all, whether it did what it was supposed to do, and also to monitor the exact deletion that may have been created. All right, so how do we detect this? We're going to PCR amplify a 500 base pair or so piece of DNA surrounding the original restriction site at the cut site. Then we're going to treat the PCR product with the restriction enzyme of interest. Right, that's going to be SAC1, R5, or SO1. Now we know what the wild type is going to look like. The wild type will be cut with the restriction enzyme because that's where the site was. And the enzyme will produce a band of predictable size. The mutant that we're looking for is not cut with the enzyme because the non-homologous end joining reaction is expected to destroy the site. So by simple restriction enzyme digestion, we can learn whether the CRISPR did the correct thing in cutting the DNA at the desired location, and we can do that prior to sequencing it. Right, so here's a baby mice, well, not baby mice, so manipulated cells, ES cells, okay, and they put a bunch of information together where they were actually injecting all three together all at the same time, not individual ones. So what they're looking at specifically is the triple mutants that are believed to be or appear to be manipulated at all the three genes, TET1, TET2, and TET3. So we're just combining a lot of information here. So the point is that the wild type sequence in all cases is cut with the restriction enzyme of interest to produce a 200 base pair DNA fragment. The successful Cas9 cutting non-homologous end joining would have destroyed that restriction site, leading to the formation of a larger product upon restriction enzyme digestion. And that's actually what you see in all of these. You actually see there's no digestion after treatment, and therefore this is first evidence that these mutants are what they're supposed to be, that have lost nucleotide sequences at the desired targeted region. Okay, notice also they're homozygous. You're not getting one cut and one not cut. It appears that both alleles were cut for all three of the enzymes in these cell lines. All right, so to verify this, some of them were sequenced, and in comparison to the wild type, for the TET1, TET2, and TET3, the in red is the PAM sequence, and immediately adjacent to that is the restriction enzyme sequence. That was the intended cut. So for the TET1 of the mutant, which is one of the triple mutants, we see as expected, we've lost a few nucleotides in the vicinity of the cut of the PAM, just upstream to it. 3 base pairs and 9 base pairs in those cases. For TET2, the result is the same, except this does occasionally lead to insertions, and in fact, one of the manipulated ones does have a 1 base pair insertion. But nonetheless, there were changes at the correctly targeted region for TET2 with that restriction site. Same for TET3. We know what the wild type sequence is, and that suffered deletions in the 2 examples that are chosen. So far so good. This is working in these ES cell lines with the expected correct and accurate targeting, efficient targeting in fact, and the production of largely small deletions at the intended location just upstream of the PAM, easily detected by a restriction digestion. All right, so looking at the sequence, all 6 are mutant, 2 alleles for each of the 3 genes, so they've all got deletions as expected. Okay, so the next step is to use the zygote injection method to inject the CRISPR components, where we're going to inject the zygote and then culture it and then transfer the embryo to a foster mother and examine what's happening. So we're going to be basically looking for the same thing in the context of a manipulated transgenic baby mouse. So injection is as before. Superovulated females were mated, zygotes collected, injected with mRNA, well, the mRNA is encoding Cas9, targeting RNAs, etc. Reimplant, and then look for baby mice. Okay, so in this case, the information we're getting is not what the restriction enzyme sequences were, but rather the overall efficiency of the whole process. Now, it certainly looks like the part where you're going from an injected blastocyst to a newborn mouse is not very efficient, right? For TET2, for example, we got 176 starting with, we transferred 108 embryos that successfully started differentiating and were cultured. And of those, there were only 22 live-born new mice and 3 dead ones. But amazingly enough, despite the loss of embryos at this stage, if we look at the number of mutant alleles present in the mouse, most of the mouse, 19 out of the 20 mice tested, were actually homozygous for the desired mutation. And that, except you could get 1, you could get 0, but in fact, it's incredibly efficient that you got both alleles in the diploid. Same for the other genes. Now, there appeared to be a problem with TET3, so they stopped working on TET3, and there seems to be a lot of lethality that out of the 15 newborns that they got, 13 were dead. So there seems to be a lethality issue with TET3, so we're going to leave that alone for now. So now we want to do the zygote injection method to get multiple mutations. So we're doing TET1 and TET2 together instead of just individually like we just did. And the results are very similar, except we do get an astonishing number of alleles of newborns that are actually mutant for all 4 alleles. We've got 2 alleles of TET1 in play, 2 alleles of TET2, and of the newborn baby mice that were tested, the vast majority had actually all 4 alleles manipulated as desired, with a few that were only singly or whatever. So this is an incredibly successful way of doing things, that you actually get 2 targeted mutations, 4 really all at once. So now we're going to try to make the transgenic mice in a way that's not going to necessarily knock out the gene, but transfer exogenous DNA to that locus by homology-dependent repair, which is much harder to do, really. It's not that efficient of a process, but it does work reasonably well. So here's our same system. We've got the SAC1 site, the EcoR5 site, and then we're not doing the zoocyte. All right, so we're going to try to do a replacement using homology-dependent repair. And what we're going to do is we're going to use TET1 and TET2, these guide RNAs that are going to be targeted to the region of interest, along with the corresponding oligos that are going to have a system that's something we want to replace the endogenous sequence with. So this is what we're hoping to have happen. It did actually happen. This is kind of jumping to the answer, that when you did the double oligo injection, we did successfully do homology-dependent repair, which is to say replacement of a sequence with the exogenous sequence at least some of the time. So that's where we're going with this. So we're going to include the homology repair templates because we hope that the cell will use them to repair the double-strand break created by Cas9, with the result that the oligo sequence will replace what's there. So this time we're again looking for something that's easily detected simply as a proof of concept principle that we can actually do homology-dependent repair in these mice in an efficient sort of way. So this is a very clever experiment, actually. Right, here are our homology-dependent repair templates that we're going to inject along with the guide RNAs and the CRISPR, Cas9, etc. So what these do is they include a non-genomic EcoR1 site flanked by sequences that are homologous to the region that we're trying to target, one for each, TET1 and TET2. So this is a very cute experiment, actually, because what we're going to do is we know that if CRISPR works efficiently, the SAC1 site will be cut because of the fact that that's underlined, that's where the guide sequence is going to target it to. So we're going to have a break in the DNA at the SAC1 site, which is either going to be repaired by chewing away some nucleotides and putting the 2 ends, ligating the 2 ends together. But that's not what we want this time. We want it to make the cut and then replace that cut sequence with the exogenous template. Well, our template now is marked with an EcoR1 site, which is only 2 nucleotides different from the SAC site at the same location. So what we're going to be doing is looking for the replacement of the SAC1 site with this exogenously added EcoR1 site for TET1. For TET2, same idea. It's an EcoR5 site. The homology template is different from it in a few nucleotides, which creates a new EcoR1 site that wasn't there in the genome sequence. So we're going to be looking for essentially the replacement of the EcoR5 site with the new site, which cuts with EcoR1. Okay, this says it's a complicated slide, and it is. Okay, so what it is the results for two genes, both TET1 and TET2, and mice that arose from this type of manipulation. So what we're going to do is just analyze the DNA from all these mice by restriction enzyme treatment in order to decide who has been repaired by homology, well, who's been successfully cut in the first place, who has been repaired by homology-dependent repair, and who has been repaired by non-homologous end joining. Okay, so the first thing to look at is the minus restriction enzyme lane with the wild type. It's the lane on the right. So without the restriction enzyme, we don't learn much except that we haven't got any big deletions, that whatever happened, happened. Right? We can see immediately that mice 3 and 9, their minus restriction enzyme site gives you a smaller band than is present in the wild type. So that says to us that mice 3 and 9 have deletions somewhere in the TET1 gene, and that we're going to have trouble believing the rest of what it is all about. All right, so the next thing which is important is the SAC1 site on the right. Look at SAC1. Okay, the wild type was cut with the enzyme, and we can see that it produces a band that runs fairly far down on the gel lane. So that is the position of a SAC1 site that was there originally. Now, if the CRISPR cut correctly, well, if it cut in the right region, the SAC1 site would be destroyed because it's adjacent to the PAM where we expect the CRISPR-Cas9 to cut. The result would be a larger restriction fragment, which doesn't migrate as far in the gel as the wild type. So we can see that most of the mice are exactly what we're hoping for. Mice numbers 1, 2, 4, 5, 6 probably, 7, 8, and 10 all have lost the SAC1 site. We can tell that because their band doesn't go as far as the wild type. So those are successful in the sense that CRISPR got there and made the cut. Now, to distinguish homology-dependent repair from non-homologous end joining, we need to look at the EcoR1. Okay, remember the homology templates contained an EcoR1 site that was not present in the genomic DNA. So when we treat the wild type with EcoR1, we can see a band on the gel which reflects cutting of adjacent EcoR1 sites to give you this band. Okay, so importantly, some of the mice got a smaller EcoR1 fragment. What that means is that the genome of the mouse now has a restriction EcoR1 site that was not present in the wild type. So we've added an EcoR1 site to the genome of these mice. And we can see that because of the faster migrating fragments that occur in, for example, mouse number 1. Well, a lot of them have it. Mouse number 1, 6, 1 and 6, 7 also, have only the EcoR1 site version with the added EcoR1 site. And some of the mice are heterozygous, implying that one of their alleles got the EcoR1 site and the other one didn't. So the ones with the EcoR1 site are those that were repaired by homology-dependent repair, because that's where they got the EcoR1 site. If the band is still the same size as wild type, and we know that the SAC1 was destroyed, that those are the ones that were repaired by non-homologous end joining. Okay, the same is true for TET2. The minus restriction enzyme tells us how big the band is supposed to be in the first place, so deviations are not allowed. Number 8 doesn't look kind of suspicious. Okay, the EcoR5 in this case, we see the wild type band, and a successful CRISPR cut with non-homologous end joining would destroy the EcoR5 site and therefore produce a larger band. So the CRISPR worked on this one. We've got all EcoR5 sites destroyed. Now, to distinguish whether that repair, since we know it cut, was by non-homologous end joining versus homology-dependent repair, we have to look at the EcoR1 again. So the wild type gives a band which runs pretty high up in the gel, and the successful homology-dependent repair products will give a smaller band because we've introduced a new EcoR1 site into the genome of the mouse. So we can clearly see that mice numbers 1 and 2, for example, 5 and 6, 8's suspicious because of the deletion, so we're going to ignore that. In those mice, both alleles were repaired by homology-dependent repair, as evidenced by the presence of this new ECOR1 site that wasn't there in the first place. We have some heterozygotes, though, like mouse number 3, for example, has one, the larger allele represents one that was repaired by non-homologous end joining, but the smaller allele was repaired by homology-dependent repair. So it's complicated, but you have to take it one piece at a time, analyze it one piece at a time, and it will ultimately make sense. So this is probably what I just said, right? Minus restriction enzyme is a control. Always look at the controls first. We can be able to see what it looks like without any enzyme. So everybody should have the same band, except for the large deletions. Okay, we see some deletions. Okay, so ignore that. That's a good thing to pay attention. Okay, so did the CRISPR work? You look at the wild type, and you look at the Sac, and you see, okay, you get a bigger fragment because the Sac1 site was destroyed. So that's CRISPR cut the site, so that means it worked. None of the DNA is cut with Sac1, meaning that at least the CRISPR part of it, the DNA was cut with a double-strand break at the correct location, etc. So for homologous, non-homologous end joining, no, for non-homologous end joining, the DNA will have small deletions, and they would therefore lose a restriction site, and they would be missing sites. But the ECOR1 would not cut because the homology repair process did not substitute the DNA like we wanted it to, right? So for homology-dependent repair, using the template, we're going to get a new ECOR1 site on that particular molecule. We have 2 alleles for each gene, so it's possible to repair one of them one way and the other way. So we're going to go through all this. This is pretty much what I just said. You'll be able to pick out the ones that were repaired by non-homologous end joining versus homology-dependent repair based on their behavior with the various restriction sites. So ECOR1 is the key for looking at the homology-dependent repair. We're going to look for the presence of a new ECOR1 site that was not there in the wild-type parental. Okay, so for a worksheet like objects, we're going to analyze the mice. We're going to look at the gel in detail, and we're going to ask which of the mice are homozygous for repair of TET1 by HDR, which are heterozygous for repair by TET1, which of the mice are homozygous for repair of TET2 by homology-dependent repair, which are heterozygous. These are the answers, but you should try to get it yourself first. I'm looking at all the bands on the gel, stepping through it one restriction enzyme at a time, etc. Okay, so now we get to our worksheet questions, and you should do these and think about them, not just take my answers for granted. Okay, so what we're just basically going to do is just what we just did. Go through and look at the mice and characterize them according to what kinds of genetic events they've had. So we're going to be able to see whether the mice that have lost the ECOR5 site, we can tell that they have or have not been cut by Cas9. We can tell whether the mice have gained or not gained the ECOR1 site, and as a result, we will be able to tell which type of repair pathway was used. We'll be able to tell who's heterozygous, and we'll be able to tell what the repair mechanism was. Okay, some more worksheet questions with another picture of our slide to refer to. So for TET1, 2 of the mice appeared to have fairly large deletions. We can tell that by looking at the minus restriction enzyme. We can tell for TET1, mice 1 and 6 appear to be either heterozygous or homozygous for the homology-dependent repair allele. And then we have some choices about TET1 and TET2, whether R1 sites are present or not, and how that relates to the repair mechanism. Right, so the answers here for TET1, mice 3 and 9 have big deletions. Mice 1 and 6 appear to be homozygous for the HDR repair allele, as evidenced by the new ECOR1 site. Combinations of TET1 and TET2. Looking at mouse 6, we can see that all 4 alleles contain ECOR1 sites and were repaired by homology-dependent repair. Do this at home. So the rest of this is basically reiterating what I just said. We're looking for the appearance of an EcoR1 site, or etc. And you need to look at the controls first, whether or not the SAC site is still there, what the fragment looks like with without the restriction enzyme, etc. So just step through this. Okay, and some of this is just extra commentary that is good to read, but not that important. I did notice that there were some submolar bands, which makes things a little, they're not equal in intensity, which can be somewhat suspicious. Might just be the enzyme didn't cut, or it might be that the mouse itself is a mosaic. Don't worry about those. So here's the mice from the double oligo injection, as promised. This is the first slide we saw, that you can see the ones that we say were repaired by homology-dependent repair. When we sequence it, we do see, in fact, a new EcoR1 site. GAATTC was not there in the original template, but it is there in the TET1 gene of mouse number 5. The other allele does not have a new EcoR1 site, and that is a deletion. So that particular chromosome was repaired by non-homologous end joining, etc. So the sequence says what the restriction enzyme told us is actually correct. Right, so whatever. This just goes through all the sequencing. You can just read this. Okay. So once the proof of concept was in place, you can do many more things, including adding epitope tags to protein sequences, add a fluorescent reporter, conditional alleles, knock out the gene entirely, etc. This is huge.